Hardy-Weinberg created a series of mathematical equations that allow us to determine the amount of recessive alleles and dominant alleles in a population. However, there are some strict assumptions to make these equations valid. So today I'd like to talk about population genetics and the ways that we can determine the alleles and genotype frequencies in a population. So when we look at population sampling, it's one of the most common ways to study human populations as well as populations of other large mammals. And so we can count the total number of individuals in a population over a given amount of years. So we can see the dynamic between different populations interacting. So a population consists of all the members of a species that occupy a particular area at the same time. So like the perch population in Wabamun Lake in 1998, the number of elk in Elk Island National Park in 2015, the number of bears in Wood Buffalo National Park in 2017, the number of people in Edmonton in 1970. So that's the population. Members of a population are more likely to breed with one another than other populations of the same species because they occupy the same location. So it's not likely that a white-tailed deer from near the Alberta-Saskatchewan border is going to breed with a white-tailed deer that lives in Banff or Jasper National Park. So genes tend to stay in certain populations for generation after generation. So we can look at population sampling of the alleles in a population, and this would be called the population's gene pool. So over time, the size of the gene pool changes. It can increase when a mutation changes a gene and that mutation survives, but the gene pool can decrease when an allele dies out due to it being not advantageous and not likely to get passed on to the next generation. So the more variety there is in a gene pool, the better the chance that the animals can survive a change in the environment. And that change in the environment could make it drier or wetter. It could change the food sources. And then the more variety there is in the gene pool, the more ability that a species has to survive that change. And this change due to environmental pressure is known as evolution. And this is the change of frequency in genes from one generation to the next, again, due to genetic pressure, environmental pressure. So we're going to use the Hardy-Weinberg principle. So the Hardy-Weinberg principle depends on 5 constants. The population must be closed, so there's no immigration or emigration. So no animals are coming or leaving that population. Random mating occurs, so there's no mating preference with respect to phenotype. There's no natural selection against any of the available phenotypes that are displayed. There's going to be no net mutations in the population, and the population must be large enough so that chance events will not alter the allele frequencies. So the Hardy-Weinberg principle doesn't really apply to populations in the wild. However, we use this mathematical procedures to determine what the allele frequencies and the genotype frequencies are in populations. But again, they don't typically occur in a natural population because there usually is some sort of mating preference if we look at like large mammals. There will be mutations over a certain number of years, and animals will enter and leave populations. So when we look at, and these are all in your data booklet, these formulas. So when we look at the equation for allele frequency, it's P, the frequency of the dominant allele, plus Q, the frequency of the recessive allele, equals 1. So this means like 100% of the alleles in a population are the dominant and recessive. So this does make quite a bit of mathematical sense. And then to look at the genotype frequency, again, this is in your data booklet, it is P squared, the frequency of the homozygous dominant genotype, plus 2PQ, the frequency of the heterozygous genotype, plus Q squared, the frequency of the homozygous recessive genotype. So we can use these 2 formulas to work out a lot of different practice problems. And we can look at these practice problems by following most of the same steps. So cystic fibrosis is a recessive condition that affects about 1 in 2,500 babies in the Caucasian population of the United States. Please calculate the following. The frequency of the recessive allele in the population. So we're going to calculate our first step is to calculate Q squared. Okay, so we're going to find out what is the percentage of homozygous recessive individuals. So Q squared, we're going to take 1 divided by 2,500, and we get 0.0004. So that is our Q squared. So our second step to find the frequency of the recessive allele is Q is equal to the square root of Q squared, which is equal to the square root of 2,500, which is equal to, sorry, not 2,500. It's equal to the square root of our Q squared, which is 0.0004, which is equal to 0.02. So that is Q. Now, our third step, we can find P. So P is going to equal 1 minus Q. So 1 minus 0.02 is equal to 0.98. Okay, so that is the frequency of our dominant allele. Now, before we can find the percentage of heterozygous individuals, we have to find the number of homozygous dominant. So we only have one unknown. So P squared is equal to 0.98 squared, which is equal to 0.9604. Okay, so that's the fourth step when we calculate Hardy-Weinberg. So we first find Q squared based on information of how many recessive individuals. Sometimes they'll give you that in the question, like if they said 40% of the population has this recessive trait, that would be our Q squared. From Q squared, we go to find Q, and then we can find P squared, and then at the end, we're looking for the 2PQ. So we're going to go 1 minus P squared minus Q squared. So I'm running out of a little bit of room here. So I'm just going to we'll go 1 minus 0.9604 minus 0.02. And our 2PQ is going to be 0.0392. And then this is the percentage of heterozygous individuals in that population. So that is 2PQ. So these 5 steps are available to do all of the Hardy-Weinberg problems. Again, we find Q squared. We can usually get that information from the question because it'll tell us the number of individuals who have the recessive trait. From there, we move on to finding Q, then P squared, and then we can find 2PQ. So Hardy-Weinberg works on an experimental level, but it doesn't really work in the natural environment because there are agents of gene pool change. So the first one's going to be mutations. And these are changes that occur in the DNA of an individual. So this would change the genes in a gene pool because an individual's genes were changed through a random chance mutation or a mutagenic substance. Okay. We can have gene flow, which is because of migration and random mating. So the net movement of alleles from one population to another due to migration. So if you took some elk from Elk Island National Park and you moved them to Wood Buffalo National Park, we'd be changing the gene pool in Wood Buffalo National Park. We also have non-random mating in lots of animal species. So random means there's no way to predict which animals will choose to mate. So like a lottery. So this is when you have things like plants or corals that will just spew their gametes into the water or the air, and just by chance they happen to fertilize one another. But even when we look at animal species, there's a lot of competition for mating, whether it's nest building in penguins, or racing up the salmon river, so the stronger salmon will get there first, or when we look at mating competitions in ungulates like deer and mountain sheep. So non-random mating is usually chosen then because of physical and behavioral characteristics. So this changes the gene pool. We have genetic drift, which is a change in allele due to chance events in a small breeding population. So if there was an avalanche in Jasper National Park and it ended up killing half of the caribou herd, we would have a change in the genetic population. Or when there was forest fires in Fort McMurray and the wolverine population was greatly affected, and so there'll be a change in the gene pool due to those events. We also have a change in gene pools due to natural selection, where individuals with greater adaptations for survival have a better chance of passing them on to the offspring. So we see this in an example of the peppered moth. So in England before the Industrial Revolution, the peppered moth existed in two varieties, a white variety and a gray variety. And the white variety was much more common because the trees there were white with little kind of brown speckles on them. However, as the Industrial Revolution rapidly changed the environment in the United Kingdom, and trees became covered with ash and soot, all of a sudden within several moth generations, so that's just several years, the population changed to be mostly gray moths and only a few white moths, because the white moths were no longer able to hide on the tree and evade predators. So due to the environmental pressure, there was a change in the gene pool. Gene pool can also be changed by the founder effect. So this occurs when a few individuals start a new isolated population. So this can happen with animals on the Galapagos Island, and we can see this happen in human populations when a small group of people go to colonize a new location. And then lastly, the bottleneck effect. So the gene pool change that results from a rapid decrease in the population size. Like I said, this can be if like a bunch of half the caribou herd was lost in an avalanche in Jasper National Park. This can lead to extinction because there's not a lot of variety left in the population. And so the population loses the ability to adapt to other changes in the environment. We can see this in larger predators in countries in South America and Africa and India, where they can be hunted nearly to extinction, and then the gene pool doesn't have enough variety to continue to adapt to environmental pressures. So that is our lesson on the gene pool, gene frequencies. I hope you enjoyed it.